All right, everybody. So this week we're doing viruses. We're going to talk about prions a little bit, and we're going to talk about vaccines because we haven't had a chance to talk about that. But viruses first. So viruses are super infectious small particles that are made of some kind of nucleic acid, either DNA or RNA, and they have proteins. And they're super infectious. They need a host cell to reproduce, though. They cannot reproduce on their own. They're really, really small. So for scale, we've got a red blood cell right here next to an E. coli, which we've seen before. And then next to an E. coli, you have viruses. They're so small. Some are really, really tiny. Some are a lot bigger. It just kind of depends. Viruses are incredibly diverse. Now, a lot of times what we will say is a virion. So viruses are playing the numbers game. They're a lot like insects in that way, where you can sacrifice one of your children because you've made, 10 million of them. And so what's the loss of one? And so viruses kind of aim at that. And so sometimes they make viruses, little baby viruses that don't really work that well. And we call those virions. And so we just call any kind of virus that comes out of an infected cell a virion, whether or not it's actually infectious, because sometimes it's really hard to tell. Now, viruses have really interesting structures. Typically, they're going to be enveloped, at least in the case of mammalian viruses. Most of the ones that we're going to be talking about have envelopes. And so what that means is on the inside, all the viruses are the same. They either have DNA or RNA in their genome. We're going to talk a little bit about some diversity that's there, but they have one of those things. Now, that's contained within a protein shell called a capsid. So it's just a bunch of proteins structurally stuck together and just forming this nice little icosahedral in this case, but they can have different structures of just this protein coat that's sort of keeping it nice and safe. So that's our capsid right here. And then a lot of viruses, especially mammalian viruses, have an envelope. And what that is a phospholipid bilayer, just like cells have, but viruses don't make their own phospholipid bilayer. They steal it from the host cell when they leave. And we're going to watch a video about how that works.
So let's say that this guy has the flu, could be any flu, and here's a droplet from his sneeze containing, if we move in and take a really close look, you see each one of those little purple things is a virus. And there are a lot of viruses floating through the air, some of which go inevitably up this unfortunate man's nose.
How did that guy feel when you ripped off half of his face?
It was interesting because we did it while he was sleeping.
Okay. I am talking with medical illustrator David Belinsky, who designed this video for Xyrus, a research company. So here comes the virus, and it's going to land on one of this guy's throat cells. So notice it's covered with little yellow knobby things. You call these keys, right?
Those are the keys.
Yeah. This is a key, this is a key, this is a key.
Okay, if the keys on this virus happen to fit the locks, which are those little purpley stick-uppy things on the surface of the cell, if there is a match, the cell, watch this, welcomes the virus in. And what's this?
This is the welcoming committee. They all interlock with each other, and they pull this membrane down into the cell.
And down it goes deeper and deeper, and that welcoming structure disperses, and the virus capsule bursts, and out comes the secret recipe for how to make more viruses in those little noodly things. So this unsuspecting cell has been tricked into guiding these virus recipes right into its own command center, the nucleus. So in they go.
And they are immediately recognized by this big pink molecule, which is a mini factory.
Yeah, what is it doing?
It threads the nuclear material, the instruction code of the virus, through one hole, and out another hole comes a brand new instruction set.
So it's a copying machine making copy after copy after copy of virus recipes, which then go out of the nucleus to little chefs, those blue peanutty things. They cook up proteins that go back into the nucleus where they are reassembled into baby viruses, and then out they go. They get covered up and head to the surface where they get new keys, and then boom! Here they come. This is an eruption of virus after virus after virus after only one virus entered the cell. But how many came out? Well, millions. Millions. So if one virus can produce 1 million babies and do it again and again and again, how come this guy doesn't just drop dead, I don't know, in like 10 minutes?
Well, because you have about 100 trillion cells.
I see. So 1 million viruses is just a drop in the bucket when you have 100 trillion cells. And anyway, remember, you do have your own immune system, which when it sees a virus usually kills it. So while the virus does multiply fast, with any luck your immune system will work just a little faster. So, yes, all viruses want to spread, that's what they do, but most of the time we do keep them in check. Most of the time.
Works. Now, viruses are incredibly diverse. I wanted to show just some examples of the incredible diversity that exists, at least structurally. So we've got our tobacco mosaic virus, which is the first virus ever discovered on the far left. You can see it's got this kind of corkscrewy looking shape. It's really weird and cylindrical. You got a typical adenovirus there with that nice typical viral structure is what you would see. This one right here with this kind of interesting triangular orientation here. You can have viruses that look a little different. So here's like an example of an influenza virus. There's its genome inside with some capsid, but then it's got this nice envelope on the outside. It would actually probably have more structure than they're showing here. And then the one that a lot of people have seen before is a bacteriophage, which this is just one type of bacteriophage, a T4. They look like little spiders almost, which is super cool. Just to give you a little more diversity too, here just some of the sizes and scales. So everybody's heard of Ebola before, and so here's Ebola right here. It looks like a snake. It's so creepy. But there's all kinds of just different varieties of viruses, and this is just a very, very small sampling of not only the diversity in shape, but also in the diversity of size. So you can go all the way down to like puppy parvo or canine parvovirus in the bottom right corner, all the way up to variola major or variola minor viruses, which that's what caused smallpox in the top left corner. Now, virus life cycles can vary a little bit, but for the most part, they're exactly the same. So they have to get into the cell, they have to take over the cell to make more baby viruses, and then they have to get those baby viruses out of the cell. And you can kind of categorize that into 5 different areas. So you got to attach, you got to actually get in there, you have to replicate, and so that's a huge process right there. So you got to make new RNA or DNA, you got to make new viral proteins, you got to get them to the right place, so that's assembly. So you have to get your new baby viruses constructed properly, and then you have to get them out. And if you're an enveloped virus, then you also need to steal some of that plasma membrane. Now, not all viruses have an envelope. There are definitely non-enveloped viruses. Those are also called naked viruses, and all they are is just that nice core genome, either DNA or RNA, and that protein capsid. So they don't have that envelope on the outside, but they are still viruses and they still infect things. So viral history is really interesting. There are tons of things that you could talk about with viral history and the kind of influence that viruses have had on human history. But we're just going to kind of zoom in on a couple of these things, some of the biggies. And so you're going to see a consistency here too with smallpox, which is variola major. And so Galen, there were some people keeping track of things back in the day, Antonin or Galen, and we don't know if it was smallpox or measles, but we're talking right around the turn of the millennium, so 165 AD, and we knew that there was some kind of transmission with armies. And so the Roman troops were coming from the Middle East-ish area, and we lost 5 million people estimated mortality. Those are obviously underrepresented numbers. They have to be bigger than that because people just weren't keeping very good track. Smallpox also popped up again in Japan. It popped up again in the New World. We don't know how many people died when the European migration came over and we brought all of our viruses to the Americas. There are estimates that there were at least as many people in the Americas as there were in Europe at the time, if not more. And something like 90-95%, some of the numbers skew that high. So estimates in the hundreds of millions and probably higher. Yellow fever is another one, not to focus completely on smallpox, but that was such a big deal. But yellow fever also caused a bunch of epidemics, killing tens of thousands of people, mostly in Africa. Yellow fever is still a problem. Now, there are more recent epidemics, HIV being probably one of the most prominent ones. So starting in the 80s, though there is some evidence of spread as early as the 30s, but you have to really kind of look for it. But a major spread in the 80s, but that was a death sentence until we were able to develop multi-drug treatment. There's still an estimated mortality of 1 million people every year, but in the tens of millions total of people that have died. And there's a lot of problems here too. So you can see in the top right corner, 1.6 million people died from TB, so that's tuberculosis, including 300,000 people with HIV. And we call those comorbidities. And when you start, there's some really interesting interactions that happen when you have tuberculosis and HIV. That's like a whole other field of study. Influenza A we've all heard of, the flu. So influenza A is the one that usually causes these global pandemics. And that's because, just like all of these other reasons, when you get large groups of people together, they can start sneezing on each other and spreading diseases. And so once you start increasing the amount of globalization that happens, if somebody gets sick on the other side of the world, think coronavirus, then they can transmit that here eventually by transmitting it between people. And so influenza A is a big one. Usually it's because of genetic shift, where influenza kind of swaps genome segments. It just has to do with the structure of influenza. But the 1918 flu, just by itself, was estimated to kill tens of millions of people. It was probably way higher than that. Ebola is another really big one that's been happening pretty recently, in the tens of thousands of people that have been killed by Ebola virus. But Ebola is really, really dangerous, which is really scary. It has a high mortality rate, but it has a really high morbidity rate, and it's really, really fast. And so you can tell if somebody has Ebola really, really quickly, as opposed to coronavirus, which we've had huge outbreaks, including SARS and MERS, before SARS-CoV-2. Now, as far as understanding the history of viruses, there's lots of people back in the day, they were trying to study things. We knew about viral diseases, we just didn't know what a virus was. And so somebody described the term virus, which is Latin for poison, to describe venereal diseases in the early 1700s. But it wasn't until we talked about this last week that Edward Jenner was able to develop the first vaccine against smallpox. We're going to talk a little bit more about vaccines today, but using cowpox. And so that was, like 80 years later. They still didn't know it was this little tiny small particle. That didn't come about until almost 100 years later. So Pasteur, coming up again, he was a very prolific researcher. So Pasteur and someone that worked with him named Chamberlain, they invented these ceramic filters. So if you've ever seen a ceramic filter, we still use ceramic filters. Basically, ceramics, you can have different size pores. And so what they did was they knew that a bacteria could not get through this ceramic filter. And so what they did was tobacco was a huge crop, and it still is. And there's this disease called tobacco mosaic disease. And they'd take the tobacco leaves and they'd grind them up, and they would send them through this filter that they knew would block every known bacteria. And so the liquid that came out of that, they put that back onto a tobacco plant, and it got the disease. And so they said, well, this must be some kind of infectious thing that's smaller than a bacteria. To be clear, it was not Pasteur and Chamberlain that figured this out with tobacco. That was Ivanovsky. He did that in 1892 and published some of his work, but it wasn't until Beijerinck 6 years later he replicated everything that Ivanovsky had done, and he coined the actual term virus. He co-opted it from that early 1700s idea of a poison, and he used that to name this new contagious living fluid that was coming through these filters. Around that exact same time, Loeffler and Frosch, they isolated the first animal virus. So they knew, hey, this disease is causing foot and mouth disease. And we know that if we can take this disease, we know there's no bacteria in there, but we can still transmit the disease. And so they were actually able to create a vaccine that way, which was really, really cool. And we're cutting ahead. So I'm skipping so much stuff too here. But there's some really interesting things. So when we first were able to figure out how to do crystallization and do some electron microscopy, we were able to crystallize the tobacco mosaic virus, and you can see it here in the top right corner. So there's an electron micrograph of tobacco mosaic virus. Because viruses are so small, we can't use wavelengths of visible light to fill the gaps between them to actually see them. So we have to use electrons and use an electron microscope to actually see between viral particles because they're so small. A couple other really interesting things, like Thieler, I mentioned yellow fever. He developed the first yellow fever vaccine, which was an attenuated vaccine. We'll talk about that. He won the Nobel Prize for that. One of the other really, really, really important things was Hershey and Chase. So Hershey and Chase figured out that it was the DNA of the virus that was infectious, not the protein. Now we know that viruses have genomes just like we do, and that is what's really transmitting all the genetic information into the host cell to take it over and actually, take over, make new baby viruses, and then, kill the cell.
In the Hershey-Chase experiment, bacterial viruses called phage were used to demonstrate that DNA is the genetic material. The phage used in this experiment consisted of a DNA molecule surrounded by a protein coat. When phage infect bacteria, they attach to the surface of the bacterium and inject the DNA into the cell. The protein coat remains on the outside of the cell. In the first part of the experiment, phage were produced in a medium containing S35 radioactively labeled amino acids. This resulted in a phage population with S35 labeled proteins, but no radioactive label in the DNA. The phage were then allowed to infect the bacteria. The phage attached to the bacterial cell and injected their DNA, but the radioactively labeled protein coat remained on the outside of the cell. The phage produced in these cells contained no radioactivity. Vigorous shaking caused the empty protein coats to be removed, but did not interfere with production of new phage in the cell. In the second part of the experiment, phage were produced in a medium containing p32 labeled deoxyribonucleotides. This resulted in a phage population with p32 labeled DNA, but no radioactive label in the protein. When the phage infected the bacteria, the p32 labeled DNA entered the cell and could be found in phage subsequently produced in the infected bacteria. This demonstrated that the DNA, but not the protein, carries the genetic information for a new generation of phage.
So now that we understand that viral genomes, their nucleic acids, are the actual infectious part of them, then we can look at how we categorize viruses. And so we base our viruses generally on their genomes, at least using a Baltimore classification. Now, viruses are very diverse. You could describe viruses based on what kind of disease they cause or what kind of specific shapes they have. But a really common way is to classify them by their Baltimore classification. The Baltimore classification was developed by David Baltimore, and it basically just looks at the genome and what their genome is constructed like. And so on the far left, you got your group 1. I won't go through all of these, but you have your DNA, your RNA, and then your reverse transcribed viruses. And so on the far left, double-stranded, so that's what the DS stands for, so double-stranded DNA is group 1. So that's just like humans and every other kind of animal where they have a double-stranded DNA, and you use that to make RNA, and then you use that to make proteins. And so you can see we're always heading towards messenger RNA. So that's the RNA that you would normally make as a transcript and then send it to a ribosome to start making proteins, because that's what viruses also have to do. Now, there are lots of other different subgroups, and this is where viruses get really, really weird. So most animals that we think about, even bacteria, have double-stranded DNA in their genomes. That's what they're constructed like. That's how they work. Viruses are not that way. They're very, very weird. And so viruses, what they like to do is they can have single-stranded DNA. They can have single-stranded RNA. They can have double-stranded RNA. They can have single-stranded RNA that they convert to DNA, which then they use to convert to RNA. Or they can also do this weird thing with positive sense and negative sense RNA. So a positive sense RNA, if it's inside of a virus, it looks just like messenger RNA, and so they can take it and they can use it to make negative sense, but they can also send it right to a ribosome and start making stuff. Okay, cool. Negative sense RNA is this really backwards way of doing things where they can take negative sense, which is complementary to the messenger RNA that they want to make. So if this has an A, then this would match it with a U, or like a uracil. If this had a G, then its opposite would be a C, as opposed to positive sense, which just has, like, it looks exactly the same. And so it's just really, really weird. Viruses are so strange, which is why I love them. Now, viruses come in really generally 2 different flavors. And so they either come as DNA viruses. So on the left here, we have hepatitis B, which is a DNA virus. Typically, there are exceptions. Typically, a DNA virus has a DNA genome that is going to enter the nucleus because that's where your DNA is, or whatever the host cell is. But we're just going to kind of assume that it's you. And so we have a nice mammalian human cell that's got a nucleus. That's where your DNA is. And so that's where your DNA gets converted into RNA, or you get transcribe it into RNA, right? And so the virus is going to send its DNA to your nucleus because that's where you make RNA, and the virus wants to make more proteins to make more baby viruses. And so that kind of makes sense, right? You have a DNA virus, so it heads to the nucleus. Now, not all DNA viruses do that, but a lot of them do. Now, on the right here, we have an RNA virus, and this is hepatitis C. Fun fact, the hepatitis viruses, there's Hep A, Hep B, Hep C, there's Hep D. They just like to infect liver cells. That's why they're hepatitis. They cause liver inflammation. They are often not related at all. Great example, hepatitis B versus hepatitis C. Yes, they both cause hepatitis, but they're very, very different cells, and they have very, very different genomes. Hepatitis C is a great example of an RNA virus. So RNA viruses, they're already RNA. So they don't need to go to the nucleus necessarily to get converted to RNA. They're already RNA. So they just stay outside the nucleus because that's a whole nother world that they have to get into. So they just hang outside of the nucleus, head into a ribosome, and get translated into a new protein, and then they can assemble. Now, there are, of course, exceptions to this as well. Influenza actually being a huge example where influenza has an RNA genome, but it actually goes into the nucleus. So, of course, there's exceptions. But for the most part, your DNA virus goes into the nucleus, and for the most part, your RNA viruses skip the nucleus and go right into the cytoplasm. Now, viral growth is really interesting. It's not dissimilar from bacterial growth phases, but it's a little bit different because bacterial growth, they don't necessarily die at the end. So when you get your virus, it infects the cell, it takes it over. It takes a little while to actually get in there and start making the baby viruses, and then you start making more and more and more and more and more and more. And then eventually you hit the point where the cell either can't contain them anymore, or it's under so much stress that it explodes. An important distinction here is that in bacteria, that's usually what happens. So if a bacteria gets taken over by a virus, it just makes baby viruses and baby viruses until the bacteria completely explodes because there's so many viruses inside the bacteria. Mammals, not so much. So what mammalian viruses like to do is they like to take over the cell, make baby viruses that head to the surface of the cell because it's huge, and then those little baby viruses will kind of bleb off. They'll go and they'll like fall off, and they'll kind of float away with that envelope with them. And then the cell can kind of rebuild its own cell envelope and just kind of fill in that gap, and then it can start over. And so mammalian cells can live a lot longer with a viral infection. However, they do typically die. In fact, it's to the mammalian cell's best interest to die so that it doesn't infect more mammalian cells that are nearby. Now, something interesting that happens only in specific families of viruses is latency. So latency is when a virus hides inside your cells, and we've heard of most of these. So if you've ever heard of herpes or chickenpox or Epstein-Barr, which causes mono, or glandular fever if you're in the UK, or HIV, which everybody's heard of, those are all latent viruses. And what they do is you get sick, and you have an acute infection, and it lasts a few days, you start to sort of clear up most of the virus, like it's kind of gone. But the trick with latent viruses is that they can take over the cell later. And so what they do is they hide their genome inside the host cell. And so I have a couple examples here. So on the left here is a proviral latency. So that would be like what HIV does, where it takes its genome. So diabolical. There's its genome right here. It takes it, converts it into double-stranded DNA from RNA, and then takes that and integrates it or inserts it into the host cell's genome. And it will always be there forever until the virus reactivates and starts making more baby viruses. But this could sit there for decades and never reactivate. It's so crazy. Hepatitis viruses do similar things, but what they do, instead of taking their genome and actually inserting it into the host genome, what they do is they can take a small like circular form of their genome and have that just kind of hang out just on the side inside the nucleus, just kind of hiding there. So it's not integrated into the host genome, but it is near the host genome. Now, that's a little bit different from persistent infections. And so there are examples of viruses, especially the hepatitis viruses, that always have a low level of viral infection. And so you could always detect a little bit of virus in someone. Even though there's not a lot of it, you can still detect a little bit. So there is still some consistent replication happening. That's different from latency. Latency is where the virus just hides forever until it gets reactivated. A lot of times I will get asked, where do viruses come from? Just to switch gears a little bit. And there are a few hypotheses. I am not an expert on viral evolution, or at least viral ancient evolution, but there are three big hypotheses, and I have a couple papers on here if you really are interested in reading about them. Nobody really knows where viruses came from. The simplest one would be that viruses came first, and that all other complex life came from viruses, but that's actually probably not the most accurate way, because just based on the genomes of organisms and how weird viruses are. And so there's more the reductive hypothesis that viruses are sort of like these leftovers from cellular organisms, and so they've kind of descended and become reductive. They've lost a lot of their abilities from when they used to be cells that could infect things, and now they're just these really simplified versions. Or they could come from host genetic elements that can move between cells, which that would be like transposons, which are definitely things that we do see in mammalian cells. And so viruses might be related to that. But for the most part, it's not really a big deal. Viruses are just so strange that we don't know where they came from. They just kind of exist. Now, to switch gears again is something that often gets talked about with viruses. They are very different from viruses, but we do need to talk about them because they're really, really small infectious things, and those are prions. So you may have heard of mad cow disease before. There was a mad cow scare when I was a kid when people were eating beef, and what would happen is people were getting really, really sick, and they would develop these holes in their brains. And so it would almost look like Swiss cheese. I have a picture in a minute of what someone's brain actually looks like when you have prion disease. And so what's happening is that you have these special proteins inside your body, but there's a lot of them in neurons. They're called prions, which stands for proteinaceous infectious particle. That's where that name comes from, which is a mouthful, I know. But basically, they're these proteins that exist inside of us, and they have a variety of functions. It's actually not entirely understood why they exist or what they even do, but we know there's a lot of them. And it turns out you can accidentally get them to sort of mutate is the wrong word because we usually use that term with nucleic acids because it's just a protein. But what you can do is get them to change their shape. And so they switch from, you can see here we have a normal prion mostly with alpha helices, which is a secondary protein structure, and then one little beta sheet right there in the red, those red arrows right here. And you can get that to switch to a huge long beta sheet with a couple of alpha helices here. But this beta sheet is what can stack, and they act just like little Legos. And so you can get them to stack over and over and over and over and over again, and they make these really, really long fibers. And what those fibers will do is they get so long inside of a neuron or any cell, but neurons are the worst, and they get so long that they actually break through the plasma membrane on either side of the cell, or at least on one side of the cell, and they kill the cell. There are lots of diseases that we know are prion diseases, especially now, but we've known of a lot of animal diseases for a long time. Kuru is the one that gets talked about in humans. That was a cannibalistic tribe in New Guinea. They would eat the brains of their elders who had died in the hopes that they would gain their memories, but it actually turned out that what they were doing was someone back in the day had prion disease, and what they would do is they'd eat this brain, and then they would get those prions, and it would start converting other prion proteins, other normal prion proteins, to the infectious damaging form. And that's what's really crazy, is that you can have this really diseased version, and if this diseased prion encounters a normal prion, it will convert it into the diseased version, and then it joins the stack. And so there are lots of animal prion diseases, scrapie being a really common one in sheep, obviously bovine, which we call mad cow disease, because what ends up happening is the cows, their brains are developing holes. So I'm going to show a picture here. So on the left would be normal brain tissue, and on the right you can see all of these holes. Those are not supposed to be there. So those are just neuronal death. And so as that starts to happen, you don't necessarily go crazy, but you do start losing motor function, just all the things that your brain is supposed to be doing, you start losing those abilities. Now, humans can acquire prion disease. You can sometimes get it from tainted meat, hence the mad cow scare. That would be an acquired infection. That's pretty atypical. Most of the time, it's spontaneous. It just randomly happens. It's super rare, though, so don't worry. It's one in a million can get it each year and actually die from it. But there's some really interesting examples of familial prion disease, which is really, really scary because people may not know that they have it, and what they will do is you don't actually suffer from the damaging effects of the familial prion disease until very late in life after you've born children. And so if you knew ahead of time, you may not have had children because they may have the disease. And so now you're stuck with it. You're like, oh no, like I have this prion disease and now my kids have it too. And so going off of that, I'd like to show this video. It's a sobering video. Luckily, prion diseases are pretty rare, but this is a sobering video of a family with their matriarch suffering from and eventually succumbing to a human prion disease.
It was just a few years ago that they were like any other family.
I've been doing a lot of thinking.
Said she still might know me, huh? I didn't think she would. He said that she just has her moments. She doesn't know yet.
I know she'd been kind of tripping a little bit, but I didn't think CJD at all.
My sister mentioned to me about the family illness that took my grandfather and my uncle. I went there, I saw my aunt, and I knew. I think she probably has CJD.
I think the hardest thing about the whole thing is I won't see my kids or my grandkids or my great-grandkids anymore. And that's what hurts.
The eyes go first, then the legs.
Everything is getting harder to do, by myself.
This is your sandwich. Yeah, big bite. There you go. Good job.
And there's the fear of the unknown, of what's to come, the fear that it's actually CJD.
I guess I realized, knowing the family history, that I was dealing with a familial illness and that it wasn't over for us. Because Mom carries the gene and it is in the family, that we have a 50-50 chance of having that gene as well.
Cindy and I had blood drawn February 1st, 99. I found out I had the gene on December 22nd, 1999.
It's 1 out of every million people get this, and then familial is 10% of that. How lucky are we? Is that hard for you to talk? Yeah. And are you frustrated because we can't hear you? Yeah.
What the family and I think is probably best at this point in time is if we keep you pretty much in bed. So
The turning point is when they're confined to bed.
A chance of another phase we're entering into, and not a very easy one.
You do feel guilty because you feel like you're giving up. You feel like you're giving away that last bit of hope. But in reality, there is no hope.
Finally, there's acceptance and letting go.
You don't have to fight this anymore. When you're ready, and when he's ready, you go to him. We don't want you to suffer.
Hail Mary, full of grace, the Lord is with thee. Blessed art thou among women, and blessed is the fruit of thy womb, Jesus. Holy Mary, Mother of God, pray for us sinners, now and at the hour of our death. Amen.
And then the cycle starts again.
I have to surrender to the fact that I'm going to see it again. She won't be the last. She might be the last in her generation, but our generation is getting up there. It's not over for us.
Oh man, that video gets me every time. So I've been wanting to talk about vaccines for a while, to switch gears once again. And this is the last thing we're going to talk about today. So 2 weeks ago, I wanted to talk about vaccines, ran out of time. Last week, we talked about the immune system. I wanted to talk about vaccines, but we ran out of time. And so this week, we're going to talk about vaccines. So if you remember from last week, when you have your immune system, when you get exposed to an antigen of some kind, then you have your primary immune response. But if you see that same antigen again, you have a secondary immune response. And it's faster. It happens much faster, within days as opposed to weeks. And the response is drastically larger than that, even the primary immune response at its peak. And so that's illustrated really, really well here by these little hands pointing at those two things I just said. So what do vaccines do? Well, they just artificially give you a primary immune response against an infectious agent. Now, there are lots of different kinds of vaccines. I have this chart here. I know there's a lot going on here. I just want to briefly talk about a couple of these, okay? So we start on the far left with live attenuated vaccines. So that's your vaccines where we take a virus and we weaken it in some way. So either that's usually through passaging. And so what that means is you just keep growing it in weird cell types. So not like a typical human cell, but like maybe a mouse cell or a mouse brain or a chicken brain. And you keep growing the virus inside those weird cells that are different from who you're trying to vaccinate. And it gets used to living inside those other cells. And you keep doing it and doing it and doing it and doing it. And then finally you take that virus that's been growing in these other cell lines and you put them back into the original host that the virus used to want to kill. And now it's not causing as much disease. And so there's lots of vaccines in that way. MMR is a really big one. Chickenpox, lots of flu vaccines are that way. Now, the other way you could do it is to grow up a bunch of the virus that wants to cause a specific disease, and then you either heat kill it or use chemicals like formaldehyde to actually kill the virus, and then you just inject that. Now, both of these ways are actually really, really effective because you still have the virus there. Live attenuated vaccines are the best because they still cause a minor infection, and that really, really bothers your body. And so it mounts a really, really good adaptive immune response. Inactivated vaccines still have all those pieces of viruses, and those still alert the body that there is a viral infection there. Even if there's not an active infection happening, it still has the pieces that would normally be there. And that really bothers the immune system. The injected polio vaccine is that way. The oral polio vaccine is a live attenuated one, but we don't typically use that, at least not in the United States anymore. Now I'm going to skip over these ones in the middle here, these replicating vectors, and skip to subunit vaccines. And so basically what's happened is our technology has improved and our understanding of the immune system and viruses has improved drastically over the years. So we've had live attenuated and inactivated vaccines since the 1800s. But subunit vaccines are basically where we can take the very piece of the virus that we know we need to make antibodies against. So in the case of coronavirus, we know it's that spike protein on the surface, and we usually call the proteins that are on the surface of viruses spike proteins. That's just what we call them. And so we know if we take those little spike proteins, just that protein, and inject that into you, your body will create antibodies against it. Now, what's really tricky is that you don't have the rest of the virus there to kind of tell the body that, hey, there's something really bad here. And so what we do is we use these things called adjuvants. And an adjuvant is just this additional thing that we use to help tell the body that something bad is there. And so what that's going to usually do is cause a little bit more inflammation, which we know pain, heat, swelling, redness, those are all signs of inflammation, and you're going to get those at vaccine sites. So if you get an injection into your arm, you're going to have all the signs of inflammation, and that's a good thing. That means that the vaccine is actually doing its job, because what you want to do is you want to get those pieces of this virus into your body and then tell the immune system that those pieces are there, because that way you can make antibodies against them. And then when you see the virus again in the future, now you don't have to, suffer a primary immune response, which can take a while, and the virus might kill you before you can even do that. So that's why we vaccinate. Just to quickly cover these other types of vaccines, because they're relatively new, is to take a virus that doesn't really hurt people much. So like an adenovirus might just cause a cold. And what you can do is you gut it, you take its genome out, and you put pieces of other viruses that you're trying to get rid of. So like a more dangerous virus, and you can insert pieces of that into this other Trojan horse, if you will, virus, and then you inject that into people. And there's a couple different ways you can do that. Either you can take, like this adenovirus doesn't work at all and inject that into a person, or it kind of replicates a little bit. But basically what you're trying to do is you're sort of replicating the idea of a live attenuated vaccine, because you have that virus there still, and it's still going to cause an infection, which really, really bothers the body and really tells it to, hey, attack this thing. And so we're sort of taking advantage of that. But some viruses are so dangerous that we don't want to make them live attenuated. And so we just take their guts out and put those into something that's not as dangerous. Now, of course, in the news are RNA vaccines, which are kind of similar to DNA vaccines. But basically with a DNA vaccine, you just take the DNA that codes for the RNA that codes for the protein, right? We've talked about that before. So you're just going to take the DNA, inject that into the host, and then eventually the host cell actually does all the work that you would normally do, like for a subunit vaccine, where you grow the protein outside the body and then inject it. Well, let's just give the body the DNA, and then it will make the protein itself, and then you can mount an adaptive immune response. Well, RNA vaccines are very similar, but we're skipping the DNA part and we're just using the RNA. And the advantages to that are really, really nice, because RNA doesn't actually last that long. And so there's all this misinformation. We'll talk a little bit about that in a minute. But there's all this misinformation about vaccines and RNA vaccines, like changing your genome. Well, first off, the DNA vaccines don't do that either. But if that was worrying you, because they could kind of stick around for a while, then use an RNA vaccine, because it's not going to stick around. It's going to generate a few of these proteins, and your body's going to be able to mount an adaptive immune response against it, because we have nice adjuvants that can really trigger that, and you can mount this adaptive immune response, and it works really, really well. Now, what gets tricky with some vaccines is that you do need to trigger a secondary immune response and sometimes a tertiary immune response just to make sure that you've done a really, really good job and maintain a really high level of antibodies in the blood. And so if I jump ahead here is a typical chart for a child. So these would be the recommended vaccines over the course of their life, starting at birth. And so you can see we have lots of first dose, second dose, third dose. And so what you're trying to do is really, really trigger a really high antibody level. Because a lot of these diseases, and people forget that, especially in the United States and in Western Europe, we forget how dangerous these diseases actually are because we haven't been dealing with them at all. So polio, for example, or measles, we really don't have to deal with those much at all, but only because we vaccinate such a large portion of our populace. Once you start dropping that threshold of herd immunity, then you really run the risk of having people develop these infections again because we have not eliminated these infectious diseases off the planet. The only infectious disease that we've completely eliminated is smallpox. And what's important, the reason you need these secondary and tertiary doses is to make sure that the level of antibodies that you actually have meets a specific threshold so that if, for example, measles virus gets inside of your body, you need to have a specific number of antibodies around to block that from actually infecting you. And it depends on the virus because some viruses are really, really infectious. Some are not as infectious. Measles happens to be crazy infectious. And so jumping off of that, I want to show this video that talks about measles and measles vaccines and why the measles vaccine in particular, which is contained in the MMR vaccine, which gets talked about a lot, why it's so important that we deal with measles.
Recently, there's been a lot of talk about measles. What does measles actually do, and should you vaccinate against it, or is this just hysteria? Measles is a virus, a hull of proteins, RNA, plus some more proteins for reproduction. It cannot reproduce by itself, it needs a host cell to do so. To understand measles, we have to understand the immune system. You might already have seen the visual system we developed to help here. Now, let's focus on the parts of the immune system relevant to measles. The measles virus enters humans through the nose, mouth, or eyes. The measles infection starts in the lungs. Measles is especially good at infecting the body's first line of defense, macrophages, powerful guard cells that protect the lungs from intruders. They enter a cell and take it over. The virus reprograms the cell and transforms it into a dangerous virus production center. Once a cell is filled with viruses, they leave the crippled cell and begin the cycle over again. But the immune system has powerful weapons against virus infections, natural killer cells. These cells basically patrol the body and check other cells for infections. If they find an infected cell, they order it to commit suicide. This is so effective that for the first 10 days or so, you will not even notice that you're infected with measles. And now, the reason why measles is so powerful. After a period of fighting and dying, macrophages alert the brain of the immune system, the dendritic cells. The job of the dendritic cells is to collect samples of intruders, travel to the lymph nodes, and then activate the heavy weapons that eradicate the infection very fast in a team effort. But the measles virus uses a dreadful tactic. It infects the dendritic cells and uses them as a Trojan horse to enter deeper into the body. The infected cells travel to the next lymph node to alert other immune cells. Once it arrives, the measles virus spreads around the virgin T and B cells and infects them. It attacks the very system that evolved to fight it. Now, things happen very fast. The lymph system spreads the virus everywhere and it enters the bloodstream, infecting cells while traveling. Measles infects organs like the spleen, the liver, the intestines, and most importantly, the lungs. The symptoms range from a very high fever, headache, sickness, bronchitis, and of course, a rash. In the lungs, the immune system was doing pretty okay, but now millions of viruses attack a second time and kill countless cells, wiping out the defense systems. In this phase, you start coughing out millions of measles viruses. Measles is so contagious at this stage that if you meet someone who isn't vaccinated, there's about a 90% chance you'll infect them. Without the protective army in the lungs, other bacteria or viruses that would usually not stand a chance can now enter the lungs and develop into harmful parallel infections that can cause pneumonia, the most common way to die from measles. Your body's immune system is now seriously wounded. Various protective systems are hurt and disrupted. The virus spreads everywhere, infecting the skin all over the body. The typical measles rash now becomes visible. And in some cases, the measles virus reaches the brain and causes a brain infection. If it does so, the chances of dying are between 20 to 40%, and there may be long-term damage. But your body is far from giving up at this stage, and it fights back aggressively. Some dendritic cells survive long enough to activate the antivirus forces of the body. Plasma cells in the lymph nodes start producing billions of antibodies, tiny proteins that mark infected cells for destruction or clump the virus together. Killer T cells flood the body and kill infected cells left and right. After 2 to 3 weeks, the body usually gets the upper hand and overwhelms the infection. But the immune system is now seriously weakened and may take weeks or months to recover, leaving the body vulnerable to other diseases. But if you make it, you are now immune. The immune system remembers the virus forever. Measles is no joke. Although 84% of all humans are vaccinated against measles, 122,000 people died because of the infection in 2014. Some people cannot get vaccinations, either because they're too young, because of chemotherapy or HIV, or because they're allergic to the vaccine. They need the rest of us to stop the disease for them. The measles vaccination is safe, cheap, and available. There are no benefits from having measles at all. You don't strengthen your immune system, and it's not more natural. Most people who don't vaccinate only want the best for their children, which is honorable. But if you ask yourself, am I putting the life of my child and other children at risk by not vaccinating against measles? The sad answer is yes. Yes, you are. Let's not play the blame game though. Let's work together and eradicate this virus. Together, we can get rid of these dreadful monsters and consign them to their rightful place, the history books.
And so that was another Kirgestadt video. Such good videos. I love their animations. Their information is so good. So just kind of ending up here, I just wanted to show this awesome GIF of herd immunity, and because that's in the news so much with coronavirus, and this is why we have to maintain a specific level of herd immunity. So just the idea that the more people that are vaccinated, the easier it is to prevent infections from going between different humans. Now that, of course, depends, as I said, on how infectious the virus actually is. So if you have a highly infectious virus like measles, you need to maintain a really, really high threshold of herd immunity. Other viruses, not so much, but it just kind of depends. Now, the very last thing I wanted to talk about was vaccine safety. So, of course, the idea of being anti-vax or being skeptical of vaccines, which I am also a vaccine skeptic, that has been around as long as vaccines have existed. And so there's some really great pictures, some really good cartoons from back in the day when Jenner was trying to vaccinate people against smallpox. And so those have always been around. People, I really like this one because it just shows a lot of the different classes of people that fall under anti-vax for whatever reason. But notice that the cliff they're walking off is the cliff of misinformation, which has always been around. So there were a couple big things I wanted to hit real fast. Vaccines are linked to autism? No, they are not. There's no connection between vaccines and autism. We could spend an entire course on that. The person to make that correlation had so many conflicting issues. He faked a lot of his data. His medical license was revoked, and he currently goes around making money by giving talks to anti-vax people. But there's no correlation. The scientific community can't find a correlation between autism and vaccines, and the anti-vax people who think that vaccines do cause autism ran their own studies and also couldn't find any link. Now, another big one is that vaccines prevent natural immunity, which I don't think people understand what natural actually means. And that's somehow leaving people vulnerable to infectious diseases. Well, no, that's not true. Vaccines, of course, prevent infectious diseases. That's like their very definition. And that's why we don't have to deal with people that have polio or smallpox or measles or all these other infectious diseases. Now, it is true, if you did get a natural infection, there could hypothetically be more antibodies against more targets. on viruses. But the problem is now you're suffering those diseases, and these diseases are really, really bad. And what a lot of these anti-vax groups fail to talk about is that a lot of these diseases, yes, your kid might not die from chickenpox. Now, the 6-month-old baby across the street, they could die from chickenpox or measles or whatever it is. But the problem is there are a lot of infectious diseases, chickenpox in particular, that can cause these long-term effects. So if you get chickenpox, you can get shingles later in life. You could have reactivated shingles go into your brain and kill you. There's some evidence that you can have increased cardiovascular problems if you have had chickenpox, but these symptoms don't actually show up until later in life. And so actually just completely preventing these infectious diseases is so important. It's not going to leave you vulnerable to them. Now, do vaccines make Big Pharma a lot of money? No. You get one dose and you don't need another dose ever again. That's not how big pharmaceutical companies work. They want you taking their drug forever. And so that's why the US government, especially with the COVID vaccine, had to say to these pharmaceutical companies, hey, we'll pay you upfront now if you develop this vaccine to remove that potential financial loss. Do vaccines cause harmful side effects? No. Now, that depends on what you define as a harmful side effect. And so that's why I say with a caveat here. So anytime you're injecting something foreign, even just the needle itself, into the body, you can cause inflammation. Now, we've learned last week that inflammation is actually really, really important, and it's what tells the immune system, hey, there's something scary here, you need to deal with that. So you do want some level of inflammation. Now, bodies are different. Some people can get really inflamed, and they don't like that. It's not fun. You have a lot of pain. Maybe your joints kind of hurt a little bit. Some people have almost no symptoms at all. So I got the COVID vaccine in my left arm, and it hurt really bad, and it was kind of swollen. My wife got the exact same vaccine at the exact same time at the exact same table, and she had absolutely no inflammation whatsoever. And so human bodies are just kind of different. Now, what can happen anytime you have inflammation, you can accidentally trigger things, but that doesn't necessarily have to be from a vaccine. That could be from literally anything. So you could stub your toe. You could get a hangnail that gets infected. You could sprain your ankle. You could get a bruise. Those are all versions of things that can cause inflammation that can thus, trigger different things in different people. And so should you completely avoid vaccines because they could potentially, 1 out of a million, make you not feel good for a little while? Probably not. Something that's been coming around, there's specifically the COVID vaccine, specifically the RNA COVID vaccine. There's some weird things going around that it could potentially cause infertility. That was a big miscommunication that was going on, or autoimmunity. So basically what happened was a couple people in the scientific community said, hey, we need to make sure when we look at the sequence, the genome sequence for coronavirus, specifically for the spike protein, which is what we're trying to target, there are some small similarities between these spike proteins and certain things like placental protein or the potential to cause an autoimmune disease. But there was no evidence that they actually did those things. That was just scientists saying, hey, as we develop these things, we need to make sure, you keep an eye on that. And of course, people did, and there's absolutely no evidence that I can find. I spent a long time today trying to find any actually published data on this. What I can find is misinformation, especially deliberate misinformation about these things, trying to sort of sow discontent. Now, the Catholic Church put out a thing about a memorandum about not taking the vaccine because it may have baby parts or fetal cells in it. So I need to explain where that comes from. So basically, what happened was in the Netherlands in the 70s, abortions were elective, they were legal. And what happened was some scientists said, hey, you're just going to throw that aborted baby away. Could you please let me have some of their cells and we will immortalize them or make them cancerous, which sounds really scary, but basically you're just making it so that the cells always grow forever. And we took those cells and they're these really special cell lines. And so what we can do is we can grow those cells forever, and there's these really nice cells, and they're nice human cells. And what we do is we test different things on these cells. So that way we don't have to test them on actual people. We can just throw them on these cells and see what happens. And so in the case of the vaccine, but also the case in basically every pharmaceutical that gets made, you test it on these cells just to make sure that they don't actually hurt human cells. Now, none of the vaccines are grown in these fetal cell lines, and none of the vaccine that would ever get injected into you actually ever touch these fetal cell lines. They just have those fetal cell lines growing in a petri dish. You throw a little bit of vaccine on there, see what happens, and then you're like, okay, this vaccine is fine. It's not like you take it out of that petri dish and then inject it into somebody. And so there's no way that these vaccines could have aborted baby parts inside of them. That's just a complete misunderstanding of what's actually happening. And so today I want to end with a nice talk from Dr. Mike on YouTube. If you've ever seen his videos, they're really, really good, great science communicator. And what he did was he walked down the streets of New York and just talked to some people about vaccines. And he's going to cover some of these things that I talked about here. But it's just a really, really nice short little video that really covers some of these big things with vaccines and vaccine safety.
Vaccines. Cue the controversy.
So to be clear, you believe that your son's autism is linked to the vaccination?
If they don't vaccinate, we could have a measles outbreak and it could become an epidemic.
You take this little beautiful baby and you pump, I mean, it looks just like it's meant for a horse.
Never has there been a safe vaccine. Never will there be a safe vaccine.
Vaccines don't cause autism, and they save millions of lives every year.
Published, peer-reviewed evidence.
So contagious.
I want to know the truth, and I'm not anti-vaccine. I want safe vaccines.
Listening to the media, you can really lose your mind trying to figure out what the real deal about vaccines is, because there's so many contradictory points. You hear people screaming all of these things, you don't know what to believe. And I empathize with the person who doesn't have a great medical education to understand what's true and what's not. We're venturing out in New York City, and we're gonna ask people what they think or what they believe or what they know about vaccines. To put it simply, a vaccine is the introduction of a virus or part of a virus that allows your body to see it, to get used to it, to build up its defense, and be protected if the real thing does come along. What do you think about vaccinations?
I don't believe in them.
You don't believe in them? How come?
I think there were like experiments, and they put the virus in us. So. Did
your doctor try and like push you or convince you to do them?
Yeah, like always. They always do that.
Why do you think they do that?
They get paid for the.
Thank you so much. Appreciate it. Vaccines are not a blockbuster hit for doctors or pharmaceutical companies. Most of the money that's dedicated to this research goes to universities.
It's as heart-wrenching as it gets, autism in children.
Well, the vast majority of medical experts insist that vaccines are generally safe.
You're going to stand on the stage and say that vaccines and autism are unrelated? It is the most bogus.
I've seen the correlations between multiple vaccines at a very young age and autism and other problems that people have.
People think autism or whatever is a risk
Buzzword, right?
That's
going around. Can I tell you like a little history about how it's really interesting? So in 1998, this guy, he was a doctor at the time. His name is Dr. Wakefield. He did this study on 12 kids, not 1,000 kids, not 1,000,000 kids, 12. Found that there was an increased risk of getting autism if they got vaccine. This specific vaccine, MMR vaccine, measles, mumps, and rubella. He was paid $500,000 to try and create this link because of a law company that wanted to make lawsuits, right? And this is only 12 kids he tested, so not like thousands or hundreds of thousands. And he found that there was an increased risk, and it was published. They took away his medical license. There was a really big deal about it. I cannot say this more clearly. There has been no association between autism and vaccination. Zero, none. If you had to convince someone that told you that despite like the best research that you've seen that there's no link to autism, that they still believe that there's a correlation somehow.
I would suggest to them that having a child with autism is a lot better than having a dead child. You know what I mean?
What I do empathize with you and understand is that the government telling people they have to get vaccines.
Right. If you don't do it, they're not to be registered in school.
Yeah. That's a great thing to talk about because in one sense, you want to have control over your body and what treatments you partake in and what illnesses you have. That's your right to choose and all of that. But on the other hand, if you don't get immunized, you can harm other people. And the reason why you put people at risk is because not every child can get the vaccine. Not every child can be immunized. Some have a genetic deficiency in their immune system where vaccines don't work on them.
In France, we have a culture pro-vaccine, but now
I've heard recently in France it's changing a little bit, and you're thinking about making it a law.
The international community stands firmly with the U.S. on this one. They believe every child should be vaccinated. And in fact, there's such a shortage of vaccines throughout the world, UNICEF says that about one child dies every 20 seconds from a disease preventable by vaccine.
I heard today, and I hear a lot of times from my patients, that they worry that their kids are getting too many vaccines all at once, or they're getting too many boosters in the first 2 years of life. And look, the important things to remember, and what I really want to reassure you of, is that that schedule is there for a reason. It's not based on a personal whim. It's not based on one person's expert opinion. It's based on data, quality studies that prove this is the best way to deliver those vaccines. I tell this over and over again to my patients, especially when I'm giving them a flu shot, that they can't get the flu from the flu shot. The reason why people start thinking that is because the season of when we give the flu shot is the most common time of year that people generally get colds and other viruses.
Just because two things happen generally at the same time doesn't mean one of those things caused the other to happen.
For those that were talking about chemicals being found in our vaccines, it's true, there are small, minute quantities of chemicals, like for example, aluminum, that stabilize the vaccine or help prevent bacterial and fungal growth. But the amount of aluminum that's found in a vaccine is less than the amount you find in baby formula or breast milk. Think about that.
Just like with everything else in life, vaccines do carry some risks. However, most of these risks are fairly minor. Redness or swelling of the injection site, a mild fever, nothing serious. There are more serious complications like anaphylaxis, which is a life-threatening allergic reaction, but it happens less than 1 in a million cases and can be fully reversible by a healthcare professional.
As a doctor, my job is to always weigh the benefits versus the risk when doing anything. You're always forced to make decisions, and a lot of the time you have to make decisions with incomplete information. But your job is to find the best incomplete information and use that to make a decision. In this case, for vaccines, we may learn something new about vaccines in the coming years. We may learn that they weren't as good as we thought, or there's some other adverse effects that we haven't thought of. But right now, the information that's available to you, the quality information, shows that vaccines are good. They have higher benefit than risk, and they protect us. So there's no doubt, vaccinate yourself and your kids.